Morning, dear students. Today we are going to start our section with the topic structure of vertebrate skeletal muscle. Why you consider the skeletal muscle to understand structure of vertebrate muscle? Because most of the studies are done using the skeletal muscle as a model to understand the structure of muscle. So here also we are going to understand important parts. First point is belly and two ends. Belly is the main portion of the muscle. In another picture, I will show you clearly. In this picture, clearly we can see that one. In this muscle, the main portion of the muscle is called its belly. Okay. Uh, Next, two ends. For all muscle, it will possess two ends. In this picture, we can clearly see that one end and the other end. That is the first point. Then we are moving to the second point, that is tendon. What is tendon? Tendon means it is a connective tissue, tough strap of connective tissue. What is its purpose? It anchor the muscle, anchor that particular muscle at each end to a bone. In the other picture, we can clearly see that this is tendon and this is also tendons anchoring that particular muscle to its corresponding bone. Okay, so that's the second point. Then origin. What is origin? Origin means it is the end of the muscle that is firmly fixed to its bone, its corresponding bone. The end of that muscle that is firmly, that is important. One end is firmly fixed to that bone. I will show you. Here we can see in this picture itself, we can see origin. This is the origin with regard to this muscle. It is tightly anchored to the corresponding bone. So that end of the muscle that is so tightly attached to the bone is called origin. Origin. Okay. Then insertion. Insertion means the other end of the muscle that is attached to the bone, but that end is free to move when that muscle contract. So against to origin, this insertion point of the muscle to the bone, that end can move when that muscle contract. Okay. Next point is regarding epimysium. What is epimysium? Epimysium is the connective tissue that cover that muscle, cover or wrap that muscle. That is epimysium. Then what is fascicles and perimysium? Like any other organ, muscle also composed of so many tissues. Tissues. We know that in the case of liver, so many liver tissues constitutes liver. Like that way, here, so many muscle tissues constitute a big muscle. Okay, so each muscle tissue inside that muscle is called fascicles. Okay, muscle tissue that constitute a muscle is called fascicle. And what is perimysium? Epimysium, we know it is a covering. So perimysium means inside the muscle, each muscle tissue, or otherwise called fascicle. It is wrapped, covered by a connective tissue. Its name is perimysium. Okay, so that's it. Then I told you that the muscle is composed of muscle tissue. That means that it certainly will contain some cells inside. Then what's the name of those cells? They are called muscle cells or muscle fibers. So that means each muscle tissue or fascicle consists of so many muscle fibers, numerous muscle fibers. And diameter of each muscle fiber range from 5 to 100 micrometers, and its length also varies many centimeters in length. And how it forms, It forms by the fusion of during the embryonic development because of the fusion of so many myoblast cells. So many myoblast cells, they fuse each other to form a single muscle cell. As so many myoblast cells fuse each other, that is why a single muscle cell possesses many nuclei. Okay. Now we have an idea about the general structure of the skeletal muscle. Uh, Next, we are going to see the ultrastructure of the skeletal muscle. If you take a single muscle cell that form the muscle tissue, that muscle cell is multinucleated. That we have already seen that it is formed by the fusion of so many myoblast cell in the embryo. So that is why it is multinucleated, long and cylindrical. What? That muscle cell. And similar to all type of cell, this muscle cell also covered by a cell membrane. The name of that membrane is sarcolemma. Sarcolemma. We can see that membrane here. This is one muscle fiber, and that muscle fiber or muscle cell, it is covered by a membrane called sarcolemma. Next term that we need to understand is transverse tubules. Transverse tubules means this sarcolemma, otherwise the membrane of the muscle cell. It has some invaginations deep into that muscle cell. That invaginations are called transverse tubule. I will show you that. Here we can see. This is sarcolemma and this its invaginations. So that invaginations are called T-tubules or transverse tubule. Okay. And what is the peculiarity of those transverse tubules? They contain extracellular fluid because sarcolemma invaginated and create that transverse tubules. That's why extracellular fluid can perclude through that invagination. So that's why this transverse tubule is containing extracellular fluid. And that extracellular fluid is rich in calcium and sodium ions. We know that sodium is the ion, is the abundant ion that we can see outside the cell in the extracellular fluid. Okay, so transverse tubule, that term also clear to you. Then next term is sarcoplasm. Sarcoplasm similar to cytoplasm. Cytoplasm means what? It is the fluid that's seen inside the cell. So similar to that, the fluid or the cytoplasm that we can see inside this muscle cell or muscle fiber is called sarcoplasm. Here we can see the fluid, that orange portion, that is sarcoplasm. And that sarcoplasm contains high amount of glycogen reserves as well as myoglobin, that oxygen binding respiratory pigment of the muscle. Okay. Then next term is sarcoplasmic reticulum. Sarcoplasmic reticulum is none other than the smooth endoplasmic reticulum that we can see in muscles. Okay. I will show you that. And this yellow branched structure, this one is the sarcoplasmic reticulum. That is a smooth endoplasmic reticulum that is possible to see in muscles. Then associated with that term, we need to understand one more term, that is terminal cistern. I will show you that. So here, this portion is shown closer here. By looking to this image, we can see this is sarcoplasmic reticulum, that blue branched structures. So, this pink one is T tubule or transverse tubule. Near to that one, that portion of the sarcoplasmic reticulum, reticulum, it is called that terminal cisternae. Terminal cisternae. This blue part of the sarcoplasmic reticulum that lies so close to the transverse tubule is called terminal cisternae. Then what is the importance of that terminal cisternae? We know that the T tubule contain high levels of calcium ions, and from here the calcium ions can reach the sarcoplasmic reticulum and can supply to the contractile filaments that we will see later. Contractile filaments during the time of muscle contraction. So that is the importance of terminal cisternae. Okay. So we have understood about sarcoplasmic reticulum, that is the smooth endoplasmic reticulum of muscle cell, and terminal cisternae is the specialized regions of sarcoplasmic reticulum that makes contact with the transverse tubules. And through that region, the calcium stored that present in the transverse tubule are released to the contractile filaments at the time of muscle contraction. Okay. Next, another two important terms, myofibrils and myofilaments. Similar to all other types of cell, here means this muscle cell also contains so many internal structures, organelles. Here, the name of that organelle or structure is myofibrils. Okay, as we look into this picture, we can understand that this is one muscle cell or muscle fiber, and inside that we can see so many myofibrils. Okay, cylindrical structures. Okay, and they are unbranched cylindrical structures seen inside the muscle cell, and that constitute 80% of the sarcoplasm of the muscle cell. What is its diameter? It is 1 micrometer diameter. And other organelles like mitochondria and sarcoplasmic reticulum, we can find them located between this, in this picture, between this myofibrils, and also associated with this myofibril we can see sarcoplasmic reticulum. Then next point is myofilaments. Myofilaments means they are certain protein filaments that forms this myofibril. Clear? We know that this is muscle cell. This is muscle cell, and inside the muscle cell so many myofibrils are present, and that myofibril even composed of again some more filament like structures. They are called myofilaments. Okay, they are and those myofilaments are of three types: thick filament, thin filament, and elastic filament. Making the entire organization of muscle more clear to you, once more I am explaining everything in a reverse order. Okay, let's see. The finest fraction, finest subunit of the muscle is myofilament. So many myofilaments, they constitutes myofibril. Myofibril. So many such myofibrils, they form a muscle cell. Muscle cell or fiber. So we know that cells together form tissue. So I am going to draw means more number of muscle cells. Another muscle cell. And as a group of muscle cell, they form the muscle tissue. What's the name of the muscle tissue? These fascicles. Fascicles. And so many such fascicles, they together form the muscle. So I'm going to draw one more fascicle. This is muscle cell. I'm going to draw one more muscle cell. So two fascicles, and for AC, I just draw only two fascicles here, and to more than or so many fascicles, they form a muscle. Okay, that is the organization in the reverse order. I think it's clear. So look here. So muscle is composed of so many muscle tissues, that is fascicles. And inside the muscle tissue, we can see so many muscle cell or fiber. And each muscle fiber is composed of so many myofibrils. And each myofibril in turn is composed of so many myofilaments. So we have seen that each myofibril is composed of numerous myofilaments, and myofilaments itself are of three types. They are thick filament, thin filament, and elastin filament. So now we are going to see about the thick filament that we can see inside the myofibril. It's a type of myofilament. Okay. So its length and diameter is 15 nanometer is diameter, and length is only 1.6 micrometer. And this thick filament, we can see this is the thick filament. It is formed of 200 or more molecules of myosin protein. Which protein? Myosin protein. That's the important thing. As it is formed of myosin protein, this thick filament is otherwise called a myosin filament. And next we need to understand about that myosin filament. Myosin protein. That myosin protein is a large protein. What is its molecular weight? Is 4,80,000 Dalton. And this myosin protein itself composed of 6 polypeptide chains. And that 6 polypeptide chains are of two types, heavy chain and light chain. Clear? Everything is it clear? Okay. Then the shape of that myosin protein is similar to a golf club. This is myosin protein, a single myosin protein. Its shape is similar to a golf club with a long tail and double globular head. So that is the shape of the myosin protein. So that type of how many myosin protein forms one thick filament? 200 or molecules of that type of myosin protein form this thick filament. Okay. And let's see. We now understood that myosin protein is composed of 6 polypeptide chains. 2 heavy, 4 light. That two heavy chains, they are helically coiled each other. We can see it here so clearly. They are helically coiled each other. That two heavy chains of polypeptide that we can see in that myosin protein. They are helically coiled each other and form the tail of the myosin protein. And in the other end, they get separated and form the head of the myosin protein. Okay, clear? So that's about two heavy chains. Then what about that four light chain? That 4 light chains we can see associated with the head itself. Okay. So 4 light chain also seen associated with the head. That's about the organization of myosin protein. It is composed of 6 polypeptides, 2 heavy, 4 light, 2 heavy, helically coiled each other. 4 light chains are there. They are seen associated with the head of the myosin protein. And such type of 200 or molecules forms that one thick filament. Then what about the arrangement of myosin protein in one thick filament? One thick filament we can see here. Those myosin proteins are arranged tail to tail manner. Tail to tail manner. So we can see that here. This is the tail. Tail of one myosin faces to the tail of the another myosin. So as it is arranged or aggregate in that way, that's why the central portion we can't see any heads. Heads are always seen outwards on the other end. So that's about the arrangement and features of the thick filament. Okay. Next myofilament that we are going to see is thin filament. And this thin filament is composed of actin protein. In the case of thick filament, it is found of myosin protein. So that filament is called a myosin filament. As this thin filament is composed of actin protein, this thin filament is otherwise called actin filament. Okay. What about its morphometrics? It is 1 micrometer long and 7 nanometer diameter. Then we need to have a close look into the actin protein that forms that actin filament. That actin protein itself consists of so many monomeric globular proteins. They are called G-actins. That actin filament itself is composed of so many globular proteins. They are called G-actins. We can see that globular proteins here. So they polymerize. These G-actins polymerize and to form one F-actin. So we can see that F-actin here. This is one F-actin. It is coiling with another one. Okay, so that polymerized G-actin units, they coiled helically each other to form one hot actin filament. Okay, so subunits are G-actins. They polymerize to form F-actins, and two F-actins they helically coiled to form one actin filament. Then we need to understand about two other types of protein, tropomyosin and troponin. In this picture, we can see tropomyosin is a filamentous protein. Where it is located, it accommodates, it positioned in the grooves of the actin helix. In the grooves of the actin helix. Then what is its weight? Molecular weight is 70,000 Dalton. And it covers, hide or mask the binding sites for myosin in the actin filament. It shut the binding sites in the actin filament that need to be bound by the myosin filament. Okay, then what is troponin? Troponin is another type of protein that we can see in this thin filament. It is a multi-subunit protein. Multi-subunit protein means consists of three subunit globular protein. And the molecular weight of these three subunits ranges from 80,000 to 35,000 Dalton. Here we can see that this is the troponin. And at what intervals we can locate a troponin protein. We can locate troponin protein with an interval of 40 nanometer. 40 nanometer, sorry, along the length of the actin filament. Okay. Then as we consider thick and thin filament, which are the contractile proteins and which are the regulatory proteins? Let's see. So, myosin and actin. Myosin means thick filament, actin means thin filament. They constitute the contractile proteins. While troponin and tropomyosin, they are the regulatory proteins. They are the securities. Next myofilament that we are going to see is elastic filament. What are these features? They are long and thin filament of 1 nanometer diameter, and it is composed of huge spring-like proteins. This is one protein, this is another protein. What's the name? It is called titin. So titin form the elastic filament. Then one more specialty is there for titin. Anybody know what is that specialty? This titin is the largest known single polypeptide. Okay, try to remember that. That can ask us some question for your competitive examination. So anyway, titin is the largest known single polypeptide. This titin, it extends from that Z line to the thick filament. What is this Z line that you will see later? Anyway, just look to this picture and understand that this titin extends from Z line or Z disc to the thick filament. Then what are the functions of this titin filament? Titin can also answer one more question. Okay. This titin filament, it holds the thin filament and thick filament properly aligned. So that is one function. And also it anchors the thick filament to the Z line. Okay. Then the third function, it acts as a spring, and by that way it gives passive elasticity that doesn't need any energy. It gives passive elasticity for the muscles. So these are the functions of the titin filament. So, well, considering the case of elastic filament, we need to understand that this filament is also a contractile proteins. Contractile proteins means those proteins that is helping in contraction of muscles. Two other contractile proteins are actin and myosin. Next thing, very important point that we are going to see is sarcomere. What is sarcomere? Sarcomere is repeating units in myofibril. We know that myofibril contains this all three different types of myofilaments. Okay, so sarcomere is the repeating units that we can see in one myofibril. This is one sarcomere, this is another one, this is another one. And this sarcomere is the basic unit of contraction. So, as a result of the contraction of each sarcomere, that is why the muscle as a whole is contracting to perform some function. Okay, and also this myofibrils are aligned or arranged parallelly to each other to form that muscle fiber. So, as the myofibril is arranged parallelly, the sarcomere in one myofibril coincide with the sarcomere in the other myofibril. That gives a banded appearance. Okay. So, next we are going to see the detailed organization of the sarcomere. For that, just look to this picture. That organization, for remembering the organization, I am giving you one code. Just try to remember. He. So, all are different bands or lines. Tahi. So, let's see what is each letter indicates. So, first one is Z. Z line. Z line, it is the border of one sarcomere. It is the wall, just like a wall to our room. This is the board. It borders or delimits one sarcomere from another one. So that is why this is Z line. Okay, now I hope that the Z line that I mentioned you earlier is clear to you. Okay, and this Z line contains alpha actin protein. Okay, and from this Z line, this thick filaments and thin filaments are arranged interdigitally, alternately. From this Z line, thick filament and thin filament are arranged interdigitally, partial overlapped pattern. So that is Z line. Then A band or A line. What is A line? That A line correctly corresponds to the entire length of thick filament in one sarcomere. So here we can see from here to here, that forms the A line or A band or A disc. Okay, and that A band or A line also contain the tip portion of the thin filament. Tip portions of thin filaments also. We can see here. Tip portions of the thin filaments also included in the A band. So A band also I hope it's clear to you. Then H band. H band, it is the middlemost portion. Middlemost portion, and that contains only the thick filament. That line or that area doesn't contain any portion of the thin filament. So this is the H band. Okay. And then one more band, that is I band. I band is that band that we can see between two A line. Between two A line. This is one A line, and this marks the end of one sarcomere. Again, another sarcomere will begin here. Okay. And from this portion onwards, again we can see another A band. Okay, so that region between two A band, that constitutes the I band. So region between two A bands, that constitutes the I band. Okay, I hope it's clear. I am going to show you one more picture. So this is Z line, and this is A band and their length of thick filament. Then H is not shown here. This is the H band. Then I extend from one A band to another A band. From here onwards, another A band will start. So that is I band. Okay. Then I'm going to show you an animation that how that muscle contracts because of the influence, because of the functioning of actin and myosin. Just an animation. Okay. So here we can see repeating units of sarcomere. This is one sarcomere. This one is another. And one myofibril is highlighting here to demonstrate how that contraction is happening. Anyway, the detailed process you will learn in the next section. Okay. So let's see. So because of the contraction of each sarcomere unit, the muscle contracts. Okay. So, by this way, I hope that the detailed ultra structure of the muscle is very much clear to you, and we can wind up today's section. In the next section, we will see the mechanism of muscle contraction.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.